Good afternoon, everyone. My name is Anurag and I'm a third-year medical student at Trinity College Dublin in Ireland. I'm going to be presenting the human ventral amygdalofugal pathway, a virtual game of hide and seek. Before I talk about this pathway, I'm going to have to give you a bit of background on what the amygdala is in a human brain. So imagine something fearful, such as a spider jumps out in front of you. What's going to happen? This information is going to travel to the retina at the back of your eye, and from there it's going to transmit to a few regions in the brain, such as the occipital lobe, the temporal lobe at the side. And the temporal lobe is where an important almond-shaped structure called the amygdala exists. When this fear-provoking information reaches the amygdala will transmit it to all other parts of the brain. But what is the amygdala involved in, and what sort of information is it passing out? The amygdala is involved in fear. So when the amygdala passes information out, it's going to be information on fear, it's going to get you to freak out. The amygdala isn't exclusive to fear, however, it's also involved in processes such as reward and motivation. Let's just look at some of the outgoing pathways of the amygdala. The anterior commissaire connects the left and right amygdala. The stria terminalis takes a long looped route to connect to the hypothalamus, while the ventral amygdalofugal pathway, which is the focus of my research, connects the hypothalamus directly. This pathway is a little bit more complicated, as it also gives off branches to other areas of the brain, such as to the basal forebrain on the left and right side, involved in pleasure, the nucleus accumbens involved in reward, medial dorsal thalamus involved in pain processing, and the brainstem, this connection which is going to be involved in arousal. So the key objectives for my research were firstly to isolate the ventral amygdalofugal pathway by tractography, which is a form of virtual dissection of MRI scans, and to also isolate each of the branches of this tract, and then to find age and sex-related differences. So how did we go about doing this? There were many steps to the process. Firstly, we got MRI scans from 90 different people, from 90 participants, 2 types of MRI scans. The first type is a T1 MRI scan, which is black and white, and looks at the different anatomical regions within the brain. That's what you can discern between. And diffusion-weighted MRI scans, which is very useful for looking at connections of different parts of the brain. The T1 MRI scan was used to generate the image on the top right, which is an analog or volume of the amygdala for that particular person. We generated that using the cortical segmentation protocol of a program called FreeSurfer 6.0. And this program, this was an automated process. What that did is it looked at previous brain atlases and used contrast differences to identify exactly where the amygdala is for this particular participant. The image on the bottom right is all of the tracts for a person's brain, and this was generated by the constrained spherical deconvolution algorithm, which we applied using MATLAB on the diffusion-weighted MRI scan. From here, the mass or volume of the amygdala was used in conjunction with the brain tracts that we have to generate the image on the right. So we have the amygdala. We know that the ventral amygdalofugal pathway has to go to the amygdala because that's one of the areas it comes out from. So we told the computer and commanded it to delete all of the tracts that don't go to this amygdala. And this is what we were left with, the image on the right. From here, we want to get further down to the ventral amygdalofugal pathway and remove extra fibers. So what did we do? We placed this green circle over here on the medial or inner aspect of the amygdala. So anything that goes outwards, which is something the ventral amygdalofugal pathway shouldn't do, they'll be deleted. And all of the parts that go inwards, which this pathway does do, should be kept in. And these are the resultant tracts. There's still a lot of fibers, but the ventral amygdalofugal pathway is embedded somewhere within this. From here, we were able to get down to the ventral amygdalofugal pathway and its branches in the end. But how did we go from here to here, where there was such a big mess of tracts that weren't this pathway, and we could really get down in the end to clean branches, the individual branches of this tract? Our results show that we could isolate this tract for all of our participants, and we could get the branches, we could delineate the branches as well. The top right image shows the branches, the nucleus accumbens in yellow, and the bottom right image shows two tracts, shows the hypothalamic branch of the amygdalofugal pathway and the stria terminalis, and it shows how both of those go to the hypothalamus. We couldn't get the branch to the medial dorsal thalamus for all of our participants, as this was inconsistently present. There in some brains, but not there in other brains. The branch to the brainstem wasn't present at all. And these two branches are limitations for our study, so I'll be discussing these in another minute in my limitations. No significant age or gender-related micro differences were found. But how do we know that our results are valid? First of all, we looked at human brain atlases to confirm this, and they complement our results. We read post-mortem studies in humans, which used many different types of techniques. This particular image on the right is from a paper by Mori et al. In 2017. The tractography aspects of these studies were very scant, firstly, and they weren't very great because they didn't identify the branches and were riddled with anatomic inaccuracies. We also read descriptions from animal studies and had discussions with academics from Trinity College for a face value of our results. There were limitations to our study, however. For example, tractography is based on water diffusivity, which means that we're inferring that all of the diffusion patterns we're getting are axons or white matter within the brain. Next, the two branches that we couldn't get, the branch from the medial dorsal thalamus and back to the brainstem. We couldn't get these because of two reasons. They're small size and other stronger diffusivity, or higher anisotropic areas nearby which diluted the effect or diluted their presence in the brain, as seen by diffusion imaging. In conclusion, we're the very first study to virtually dissect the ventral amygdalofugal pathway and all of its branches in living human brains. And our study demonstrates how neuroimaging and VR techniques can synergize to inform neuroanatomy. Our study can open avenues into research into this bundle in various pathologies. Major depressive disorder being the one I'm going to be studying after having found this tract. Here are the references I used throughout my presentation, and I'd like to thank you all for listening, and I'd like to especially thank my co-authors for all their hard work and effort into this study.